initial housekeeping. Here we are. Welcome to my talk. This is an update on PCR and I-SPY 2 in neoadjuvant breast cancer treatments. This is a talk I gave at the Oncology Biostatistics Seminar at Johns Hopkins, November 16th, 2021. So as an overview, I'm first going to review a previous talk I gave on PCR and neoadjuvant breast cancer several years ago, and then PCR prognostic factor. Is it a biomarker, a surrogate? We'll get into that. And then the I-SPY 2 design as it was originally designed in 2002, and then plans for I-SPY 2.2, which is the basis of this talk. And we're going to talk about how does this fit into precision medicine and the minimization of chemotherapy. And then we'll look at additional prognostic factors that will be included in the design of I-SPY 2.2 and the role of the Data Monitoring Committee and the seamless transition to phase 3. And what about the contemporary control group? Is it a misnomer for historical controls? And how acceptable is this as a control group? Frequentist issues, multiplicity will be covered, and then other phase 3 design issues. Now, back in 2014, I gave this talk before the Oncology Biostatistics Seminar, and I defined PCR at that time as the lack of all signs of cancer in tissue samples removed during surgery or biopsy after treatment with radiation or chemotherapy. Now, that was an internet definition. Oncologists have a more technical definition, but for our purposes, this is sufficient. In both 2014 and then revised in 2020, the FDA issued a guidance for industry on pathologic complete response in neoadjuvant high-risk early-stage breast cancer. And it can be used as an endpoint to support accelerated approval. Well, why did we need this? Well, for DFS, disease-free survival, or EFS, event-free survival, or overall survival, it would usually take 10 years of follow-up to yield the required number of events. And pharma companies were not developing drugs for this space. So there were 2 completed NSABP trials that showed some evidence of correlation between PCR and OS. And FDA issued a guidance indicating that accelerated approval could be granted using a PCR endpoint, but a confirmatory trial would be needed. Now, here are the 2 NSABP trials I was talking about, the B18 and the B27, and you can see for OS, the hazard ratio of PCR to those who did not achieve a PCR is 0.32, highly significant, very similar in the B27 trial. Then a meta-analysis of 14 trials was done where there was a hazard ratio of 0.48, again, showing very good advantages to achieving a PCR for as a prognostic of OS. And in the Lancet in 2014, Cortazar published this paper, Pathologic Complete Response and Long-Term Clinical Benefit in Breast Cancer. 12 international trials, 11,955 patients, and the association with long-term outcomes was strongest in the triple-negative breast cancer and the HER2-positive patients, receptive negative tumors, who received trastuzumab. However, overall, there was an association. But it is a prognostic factor. It's not been validated as a surrogate biomarker. This can only be done by considering trial level data, not individual patient data. The question is, for surrogacy, is the treatment effect on EFS and DRFS, that's event-free survival and distant recurrent free survival, is it mediated through PCR? That is, if I can control PCR, then I control EFS and DRFS. So here is what Cortazar did. Looked at the trial level for these 12 trials. For EFS, the x-axis is the odds ratio for pathologic complete response. And the y-axis is the hazard ratio for event-free survival. So if there is to be a correlation, we would expect these bubbles to all be on the line, and we would expect the line to have a significant slope. And here it is for overall survival. Again, the bubbles are not exactly on the line, lots of variation, and also there isn't much of a slope in that line. The R square in this analysis for EFS was 0.03 and for OS 0.24. So again, there really is no meaningful association. Now, the Electoral College will be eliminated before PCR is validated as a surrogate, at least by this definition. Most likely, if that were to be done, the R-square lower 95% confidence interval boundary would probably have to be greater than or equal to 0.90 for people to be convinced, for it to be persuasive. But additional studies can always be published with a low R-square, even for specific subtypes, so that if a researcher were to show 8 trials and the R-square is above 0.90, then another person can come along and say, well, some of those trials had different eligibility requirements, they need to be thrown out. Or here, I have 3 new trials, add them, and now the R-square is lower than 0.90. So that this definition would not be a once and for all persuasive definition of PCR as a surrogate. No doubt it is a prognostic factor. Then AbbVie, people at AbbVie did simulations back 2014. They modeled PCR and OS to find the practicality of a confirmatory trial, that is, after the accelerated approval, and that the correlation between PCR and OS was bounded above at 0.707. That would be a 49% R-square, and the correlation is moderate at best. So, discussion, again, this is of the 2014 results. Moderate correlation, the success of a confirmatory study following the establishment of a PCR benefit to confirm the drug effect is heavily dependent on the hazard reduction of PCR to non-PCR and the treatment effect in PCR, that is, treatment versus control. And in general, the sample size required for the confirmatory trial is large, and the trial duration could be very long. Well, ODAC, that's the advisory committee at FDA, in September of 2013, at this meeting, there was uncertainty regarding PCR as a surrogate No U.S. patients were in the trials analyzed. Chemo that was used is not used in the U.S. And the chemo sequencing is different than what would normally be done in the U.S. But nevertheless, the panel voted 13 to 1 for accelerated approval on the basis of pertuzumab's performance in the Cleopatra trial. And this was a trial for metastatic breast cancer, not for early stage high risk. And eventually, the FDA approved pertuzumab without a confirmatory trial on the basis of what they called the totality of evidence. The confirmatory trial would be difficult to do, FDA admitted, enrollment, crossovers, length of time, etc. And however, even though FDA conceded that, they said they would not excuse other sponsors from a confirmatory trial. And this created tremendous controversy. Now, in JAMA Oncology in 2020, what is now known as the I-SPY2 Trial Consortium, published this paper, again looking at the association of event-free and distant recurrence-free survival with individual pathologic complete response in a group of trials. So the considerations were the neoadjuvant trials that they considered was a taxane plus an investigational agent. After surgery, doxorubicin, which is denoted as A because it was once known as adriamycin, and cyclophosphamide C. This was the protocol. And the oncologists want to eliminate post-surgery traditional chemo whenever possible, and that is now a goal of I-SPY 2.2. So the individual evaluation of PCR, they would evaluate if the correlation of PCR in high-risk operable breast cancer is associated with EFS or DRFS in the neoadjuvant trials of I-SPY2. Now, in doing this, many subtypes are considered, and many SOCs, standard of cares, in the control group have to be considered. But they looked at individual level PCR and 3-year EFS, DRFS, I-SPY2 trials from 2010 through 2016 with follow-ups to 2019, 950 patients, 330, 34.7% achieved a PCR, and 95% of those who achieved PCR also reached 3-year EFS, DRFS. And the trial consortium considered this persuasive. Let's move ahead with a new trial. And here are the hazard ratios they found for EFS, 0.19. And these are hazard ratios in favor of PCR, of course, and they're low as they have been before, much in favor of PCR. And this was similar across all molecular subtypes, which now can be considered in 2020. That was all experimental back 2014, and novel treatments that got into this analysis. And PCR is associated with an 80% reduction in recurrence rates. Lots of evidence for PCR. So as a result, I-SPY2 is a PCR-based phase 2 trial. It's an adaptive pilot program, umbrella trial, seeking new treatments for stage 2 or 3 breast cancer patients with high risk of early recurrence. Began in 2010. Bayesian posterior probability of success is in a proposed similar standard phase 3 trial would have to be greater than 0.85 for an agent to be successful in I-SPY2 and progress to phase 3. Now, again, I just mentioned Bayes. The I-SPY2 has always been and will continue to be Bayesian posterior driven. Now, 12 agents to date have completed accrual and 7 graduated to phase 3 in at least one tumor, subtype. And I show here an example of a subtype. This is not particularly significant here. I just want to show the notation that pathologists use for subtypes. So the flow chart for I-SPY2, it's an adaptive process. Several agents from several sponsors are entered, and they begin trial with equal randomization probabilities. Now we calculate the success probability for each signature, each subtype. Now, this is done with Bayes, with the Bayesian posterior I said before. Now, eventually, does this graduate or go to futility? If the answer is yes, we stop accrual in that arm. So it's important to note that we can stop the trial for either good news or bad news. Good news, it graduates to phase 3. Bad news, futility, it's not going to graduate. Well, okay, if it does neither of those, we revise the randomization probabilities within each disease subtype so that the higher the success probability in the posterior, the more patients will be randomized to those arms. And we continue the trial and we continue to calculate the success probability for each signature. And then we go back again, graduation or fertility, and this goes on. And you see in the upper right-hand corner, the accrual rate permitting, we add experimental arms as sponsors are now contributing new candidates. And there may be 5 or 6 experimental arms working at the same time. The I-SPY website, here is their the link, and it is very good for learning more about this and learning about the history of I-SPY2 since they began randomizing patients. Now, this article appeared recently in an ASCO publication, I-SPY 2.2, and I was interested in this. It's by Laura Esserman. She is an oncologist who is the chair. She's at UCSF, and she's the chair of the I-SPY trial consortium. So the goals of I-SPY 2.2, there are several, but improved tumor classification that they can do today allows for improvement in PCR prediction and will allow for improved optimal agent assignment, that is personalized medicine, for this molecularly high-risk population. But now, in I-SPY 2.2, they're going to consider additional prognostic factors, residual cancer burden, and circulating tumor DNA, ctDNA. Residual cancer burden is estimated from routine path sections of the primary breast tumor site and the regional lymph nodes after the completion of neoadjuvant therapy. 6 variables are included in the calculation formula. The calculated RCB index value can also be categorized as one of 4 RCB classes. So here is an example of the calculation. So these data are entered for each section that they get for each patient, and then they have an RCB. ctDNA is it's a circulating tumor DNA that is found in the bloodstream and refers to DNA that comes from cancerous cells in tumor. Of course, most DNA is inside a cell's nucleus, but as a tumor grows, cells die and are replaced by new ones. The dead cells get broken down, and their contents, including DNA, are released into the bloodstream. So there are small pieces of DNA. if we analyze the genome of these tumor cells using ctDNA can help doctors determine which treatment will be most effective. So this gets thrown into the models as well. So the goals, I-SPY 2.2, everything is Bayes driven. For each regimen, use PCR to predict EFS and DRFS. Predict RCB as soon as they can after each regimen but before surgery. Use ctDNA to predict residual disease. And the optimal point, what is the sweet spot between PCR but at the same time a low RCB? And that is the goal. And they want to avoid chemo, doxorubicin, and cyclophosphamide if possible, if the data shows that can be done. The degree of response determines the dose and the need for chemo, as I just said. Taxane combination is added only if necessary. And use state-of-the-art lab procedures to further match tumor type to drug class. They want to get 90% of patients to PCR and a seamless transition to phase 3. And then in 3-year follow-up, 92% EFS and DRFS. Now, several members of this consortium have said perhaps a lower PCR, but a better quality of life and less toxicity. Not clear that FDA will approve this depending on how low the PCR goes, because FDA believes in the PCR. Now, the I-SPY 2 design is in Block A, Block B, Block C, and I will show a diagram shortly. But what we do is, in Block A, test the most promising novel agents without standard chemo, but then we have a targeted de-escalation. Patients who are predicted to have a complete response after Block A proceed immediately to surgery. However, patients with poor imaging response at 3 weeks will repeat imaging at 6 weeks and proceed to Block B if response is not identified. In Block B, patients proceed to the optimal treatment combinations, that is, what is recognized as the best in class for each subtype, based on results from I-SPY 2, previous trials, and other practice-changing trials. Then Block C, patients with persistent disease after Block B will proceed to chemo, and those with predicted complete response will proceed to surgical resection. Well, it's easier to describe this with this graph. First, we do an adaptive randomization based on subtype, again with adaptive probabilities of assignment. And then let's say there are at this time 4 experimental treatments contributed by sponsors. And we try to predict the RCB at this point. If there is no response, then we go to another adaptive randomization, which I'll describe in a moment. But let's say the patient comes out of one of these experimentals, and now we predict the RCB. We can predict it. If it is a satisfactory one, that is a 0 or 1, we go to de-escalation and the patient goes right to surgery. But let's say the RCB is 2 or 3, less favorable. Now they go into an adaptive randomization based on subtypes and biomarkers for the best in class. Now, these are not the drugs that are currently in the I-SPY 2 trial. This is what we've learned from the past to be the best in class for these types of patients. Um, if they now they emerge from that and we get a predicted RCB again, if it's good, they go to surgery. If it's still not favorable, that these are the patients that will get the AC. And there is a regulatory approved standard of care is throughout the control groups that are used. And indeed, the New York Times had an article recently indicating that some breast and lung cancer patients now can be treated without chemotherapy at all. And that's referring, at least for breast, to what can be achieved through this I-SPY 2.2 design. So to discuss this, these are very ambitious goals. I think the oncologists have added considerable operational complexity to I-SPY 2. Can this complexity maintain excellence over the 28 I-SPY 2 sites? And it's not clear what lab work is centralized and what is local. This is high-tech lab work that we're talking about. And it would be nice if it could be centralized, but it's not clear that that will be the case. And if it's not, will this lab work be uniform quality across 28 sites? Well, what about the I-SPY 2.2 operating characteristics? Well, there are no operating characteristics because this is not a frequentist trial. Bayesian posterior probability distributions of PCR are regularly updated as the trial progresses. Again, a successful agent will have a posterior probability of success in a hypothetical 300-patient phase 3 trial of greater than or equal to 0.85. And however, decisions like this, all of the decisions of progressing and that I've mentioned before, are made on the basis of a small sample size as patients are recruiting. And this is open label, it's phase 2. Open label is not unusual in phase 2, but we have to consider it's also open label. Now, what about this control group? They're using contemporary controls, not concurrent. Now, what that means is they're constantly getting patients treated, being randomized to SOC. So why throw out the ones from the past? So these are essentially historical controls, but they're adjusted for time trends using the time machine. This downweights patients observed further back in time. And the drift in prognosis, that is PCR of patient population over time within subtype, is corrected for using an offset logistic model. However, with these control groups, the sponsor is suspicious of the result for their drug because they're not sure what the control group consists of at that moment. However, supposing they do, supposing they know what the control group is looking at this time because they had another drug in the I-SPY 2 last month or 3 months ago. Well, then, considering if the control group is at a random high or random low, that may affect the sponsor decision to enter the trial. So if the control group is patients are doing well, it's currently at a random high, a sponsor might say they're not going to enter their candidate. If it's at a random low, there would be a rush to enter the candidates. It's sort of like the stock market. Now, adjusting for time trend, this is the time machine I mentioned. I might just say that this began in bridging different eras in sports. It was originally developed for sports and mainly to be broadcast on the sports network ESPN. So, and the first application was the baseball home run records. Mark McGwire set a record in 1998. and beat Babe Ruth's record of 1927. Well, how do we compare 1927 with 1998? There were differences in rules and the length of the season, the nature of the ball, etc. Well, if you consider network meta-analysis, not network meta-analysis of sports, but of clinical trials, if 5 years ago there was a treatment A versus treatment B, and 3 years ago there was a trial, treatment B versus treatment C, we can do a meta-analysis network combining those and come up with an A versus C. Well, as an analogy, if we considered Babe Ruth in 1927, the linking person it would be Roger Maris in 1961, that is the B above, and then Mark McGuire in 1998 is C, then that's what they did. However, in their actual analysis, they considered more players between 1927 and 1998, just like the time machine considers the continuum of SOCs from 2010 to the present. Well, not everyone was excited about this. These three authors had a paper in this year's New England Journal. Platform trials, beware of the non-comparable control group or the non-contemporary control group, and they come down pretty high on that. These authors also say, well, look, we're going for precision medicine here. We need randomized trials, but also concurrent controls. And, well, the frequentists step forward and say, I want to know about type 1 error inflation in the presence of a trend, a linear or step trend. And these authors have investigated that. But what about the frequentist properties for a non-Bayesian platform trial? Hypothesis tests are not independent because of concurrent controls. Do we adjust for multiplicity? Should we be computing the family-wise error rate and the false discovery rate? But it is also said that each sponsor is making their own hypothesis test. So no adjustment is necessary. We're not doing an experiment here of the five treatments that happen to be live now in I-SPY 2. Rather, each sponsor has his own one drug in there and is making their own hypothesis. And there was a paper in Clinical Trials where these authors have said no adjustment is necessary for that reason. And then Clinical Trials invited these authors a commentary to see if they might disagree. And in fact, they not only agreed that no adjustment is necessary, but they even gave a stronger argument. Well, what is the role of the DMC? Well, we tried to find out, and nobody seemed to know how a DMC would operate. But I go back to my book, second edition in 2017. The challenge for DMCs with the umbrella design is that being a master protocol, the trial never closes. So there will have to be a rotation of DMC members, preferably with interlocking terms. There will be limited safety information from some drugs that don't last on the protocol very long. But DMCs will be very important in making recommendations on those drugs being elevated to phase 3. Perhaps one member of the DMC should follow that drug to chair the phase 3 DMC. That would be a good idea. And DMCs will also advise when enrollment cohorts should expand or cease and decide when to release data and to which parties, as they usually do. Anyway, that was my take on what a DMC might be faced with and how they might operate in I-SPY 2.2. Well, what is the control group for phase 3. Now, again, this isn't going to be anymore the time machine. This is a new concurrent control group for the phase 3 trial. Would control patients be treated by an SOC? That is, receive the chemo that Bayesian models in phase 2 have concluded are not necessary. So let's say for a certain subtype, uh, we find escalation to include chemo is not necessary. So in the phase 2 in I-SPY, we did a de-escalation. Well, nobody knows at this point. Again, would it be ethical to make this subtype go through chemo when the phase 2 said it wasn't necessary? Yes, that maybe is an easy answer. But then if we're testing, comparing a de-escalated treatment with the standard of care, which includes chemo, there's more than one difference between the two treatment groups. They don't differ merely by treatment. And that might be an inference problem and not lead to persuasive evidence. Anyway, nobody seems to know at this point among the people we asked. What are the estimands for the phase 3 trial? That is a new component of our protocols, but this needs to be defined. Are we making decisions on confirmatory trial designs too quickly? Are we moving too, is this too mechanized? At the time we arrive at phase 3, do we really know the dose and the safety of this trial? Will sponsors proceed to phase 3 when the subgroup where efficacy was found is a small market? I hate to use the term market, but let's face it, the sponsors are big pharma mostly. And if they get a success out of phase 2, oh, damn you. If they get sorry, sorry for that interruption. There was a phone call came in. I should have turned that off. But so again, nobody seems to know whether there'll be exactly what the control group would look like. Now, okay, let me start over. What are the estimands for the phase 3 trial? Well, we are, again, the estimands, not only is this new and has to be included, but is the Bayesian models for the posterior probability of success, is that using an estimand? Is it the treatment policy estimand? Is it the while-on treatment? Is it a composite estimand? These have to be answered. And again, will the sponsors proceed to phase 3 when the subgroup where efficacy was found is a small market? Again, being big business, they might not want to go ahead if there are very few patients of that subtype out there. Will future patients prefer to enter the phase 3 trial rather than the phase 2 I-SPY 2.2? So now we get future patients in a subtype, and there's a phase 3 trial going on. Will they volunteer for the phase 2 trial when they can go into a phase 3? And is the type 1 error for progression to phase 3 still of interest? That is, are we, what are the errors in the decision for progression? Is it not of interest anymore because of Bayes? Or do the frequentists have a point here? Also, what is the role of AI and machine learning can that be combined somehow with the I-SPY 2.2 results? And what about principal stratification for PCR as a surrogate? Has anybody tried to use the principal stratification causal inference method to validate PCR as a surrogate? And what about the cost of these drugs? And how do we get a clear idea of risk benefit? Again, success in a phase 3 trial is achieving a certain EFSDR, FS, but what about safety? The future of drug development for breast cancer lies in the neoadjuvant setting. More work is needed to sharpen this predictive tool, but statisticians must be involved at every level of this process. Thank you.